Okay, so good morning, everyone. My name is Samantha Case. I'm a fifth-year graduate student with the P2EP. I'm the graduate mentor for the Blueberry Metabolome Mapping Group. I'm part of the Department of Plant and Microbial Biology, and my advisor is Dr. Julie. So before the interns get into their project, I'd like to introduce them because without them, I wouldn't be this far in my thesis, and I wouldn't have so much great data to work with in the future. We have Alexa from Catawba College. We have Roa from the University of North Carolina at Chapel Hill. We have Anjali, also from the University of North Carolina at Chapel Hill. And we have Akola, also from Catawba College. Without further ado, Alexa will introduce the rest of the projects.
So over the summer, we've been looking at blueberries, and North America is actually the largest producer of blueberries in the world, and North Carolina is ranked number 6 in the nation. So over the past 20 years, consumption of blueberries has increased by 402 billion pounds in just the US alone. And now, thanks to research from scientists, blueberries can also be grown in Europe and Asia as well. So blueberries offer a whole bunch of health benefits, but because of their potent antioxidant capacity, antioxidants donate an electron to stabilize free radicals that are in the body that damage healthy cells by taking electrons from them. So this reduces oxidative stress, which one of the oxidative stresses is inflammation. So this reduces the risk of chronic inflammation and heart disease. Blueberries also slow the loss of cognitive function. So eating a diet full of blueberries can also reduce the risk of Alzheimer's disease. And blueberries are also ranked number 3 in dietary fiber after plums and prunes. So over the summer, we developed a non-targeted metabolomic profile of our blueberry sample extracts. And we conducted association mapping to determine genomic regions in different traits. So we can give these genomic regions to farmers so they can selectively breed for the most nutritious and the most consumer aesthetically pleasing berries.
Okay, so we started off by freeze drying 224 different blueberry samples in order to concentrate and stabilize our samples. Then we extracted the metabolites from the blueberries into a solution and acidified the extracts to further stabilize the metabolites. With our blueberry extractions, we have ran an optimized DPPH in order to evaluate the antioxidant potential of each of the blueberry extractions. Furthermore, we have conducted a non-targeted metabolomic profiling using protein NMR and HPLC QTL MS. So the protein NMR was used to identify the primary metabolites within the blueberry, such as sugars and amino acids, while the LC-MS was used to quantify specialized compounds like phenolic acids. Finally, we used the DPPH results and the non-targeted metabolites to perform an association mapping. So the DPPH is a simple, rapid, and inexpensive free radical scavenging method. DPPH is a free radical, so the assay measures the extracted compounds, how it quenches the DPPH. So the DPPH method for our blueberry extractions was optimized using Trolox as the standard, which is a natural antioxidant. And throughout the summer, consistently, we have produced over 30 Trolox standard curves with an R2 value greater than 0.98. This indicates a very well-optimized sample compared to the ideal standard curve R² value of 1. So the figure to your right is the Trolox standard curve that we have used to calculate the Trolox equivalence or the antioxidant capacity of the blueberry samples. As you can see, the distribution of the samples, which is indicated by the purple circles along the standard curve, shows the variation between the samples. And this variation is important for the association mapping.
Okay. So for association mapping, the first thing we had to do was look for quantitative trait loci, which are QTLs. These QTLs show significance in genetic markers within the variants of the traits. So for our purposes, we set a threshold value for our LOD, which shows significance of 2.5. In figure A, we found this graph from linkage group 10, And this shows we have many genetic markers that are above our threshold value of 2.5. So we can say that markers in linkage group 10 are significant, versus in figure B, there are no markers that are above our 2.5 threshold, so we can say that linkage group 11 has no significance. So the way that we come up with these QTLs is by running interval mapping, so IMs and CIMs. IMs have a high chance of false positives, but they are a much faster process, whereas CIMs are a very much longer process, but it runs through permutations of the IMs. This way we can reduce false positives. However, to prevent overcorrection, we use the data both from the IMs and the CIMs. So we compiled the IM data and the CIM data from all of our DPPH data, as well as data from over 200 different metabolites. Then we formed them into a QTL map, which maps the significant traits within different linkage groups. Linkage groups, in this context, relates to different chromosomes within the blueberries. In the figure to the right, you can see that there are 12 different linkage groups that correspond to the 12 different chromosomes in the blueberries. So after we have mapped these traits on these QTL maps, we can then screen for desirable traits and phenotypes in the blueberries and then selectively breed them to form the ideal blueberry. This is a QTL map that we came up with this summer, and it maps antioxidant capacity, or the DPPH, against some of unknown compounds we identified through proton NMR. The open red circles are indicative of the DPPH, or the antioxidant capacity. There are multiple different markers in each of these regions because we have used DPPH data from both 2 years and 2 different locations, so we can ensure that these QTLs are very accurate. After we have mapped the DPPH data, we then map the unknown compounds over top of it and look for overlaps in the data. As you can see here, unknown compounds 124 and 125 do overlap with DPPH in 2 regions in linkage group 10 and 12, so then we can foreshadow that maybe 124 and 125 do contribute to antioxidant capacity in blueberries.
This brings us to consumer appeal. So we found that consumers are less likely to consume shriveled small blueberries and are more likely to gravitate towards the plump, luscious blueberries. So we've been looking at a couple consumer traits such as taste, color, and texture. And taste, we found through research, has been composed of fructose and glucose as the main sugars that contribute to it. And then color is made up of the anthocyanins within the skin. And then texture can be broken down into 2 categories of firmness and crispness. Firmness is often measured using instruments and can be defined as the force needed to obtain a given deformation. And then crispness is measured using sensory panels and is often related to the way a grape may pop in your mouth when eaten. So we looked at a couple different traits versus our taste traits. So we looked at sugars and acid metabolites. And so in the orange box, we see acid metabolites overlapping with pH and titratable acids. This gives us confidence in our QTL mapping because these are traits we would expect to overlap. And then in the green boxes, in linkage group 8, we see an abundance of sugars overlapping, which suggests that this region may be in correspondence with a sugar synthesis. And then in linkage group 12, we have a combination of sugars and acids overlapping with pH, which may suggest that a sugar-acid ratio contributes to the overall pH and taste in the blueberry. Then we also looked at unknown compounds in comparison to taste traits. And in linkage group 12, we see unknown compound 99 overlapping with pH and titratable acid and soluble solid ratios, which suggests that this may also contribute to the acidity or pH in the blueberry as well as taste. Lastly, we looked at other consumer aesthetic traits and other unknown compounds. And so we looked at amino acids and unknown compounds. So in the orange boxes, we see amino acids overlapping with berry firmness, which suggests that these amino acids may contribute to the tension in the berry skin, producing a more firm berry. And then lastly, we see unknown compounds overlapping with percent juice, which may suggest that they may have some relationship to water retention in the berry.
So obviously these interns have created a lot of great work for me, and they've done a lot of great work. But this means that now I have a lot of future work ahead of me, and I'm really excited to get started on it. So first thing I'm going to do is start identifying some of those unknown compounds they found. So unknown compound 124, 125, and 99, because those are clearly really important for taste and antioxidant capacity. So I do that probably with utilizing more of the 2D NMR as well as more LCMS fragmentation. In conjunction with that, I would also like to look at identify more of these putative candidate genes underneath those QTL regions that may be regulating some of those traits. So I'd use a combination of the text mining techniques at UNC Bioinformatics that'll be went over later, as well as pathway analysis, so I can figure out how those things are being regulated. And then I would take those candidate genes and functionally characterize them so I could figure out the mechanism of regulation so that way we can breed for those traits. So in conclusion, blueberries have a plethora of health benefits. They are great for reducing oxidative stress as well as gastrointestinal health, improving that. So we use the combination of metabolomics and genomics to identify these overlapping QTLs that can be further used to breed for better blueberries. We want to breed for improved nutrition, better consumer aesthetics, and higher productivity, so therefore we can provide so more people can eat all of these blueberries, so therefore everybody benefits from the health benefits in blueberry. So obviously there's been a lot of people that have helped me along the way. I'd like to acknowledge them all because I wouldn't be here without them. So the members of the Lee Lab, they've helped me with the metabolomic profiling. The Lila Lab has helped me with the antioxidant capacity assay as well as extractions. The DHMRI, NMR researchers, they've helped me with the NMR data. The UNCC Bioinformatics Services Division, they helped me with sorting and storing the data. General Mills Crop Biosciences and Berry Blue has helped me with some of the taste panel phenotypes as well as some of the genetic information. My fellow graduate students, you guys have helped me so much along the way, and obviously I wouldn't have gotten as much done without my P2EP interns, both the past and present interns. So come ask us questions at our poster. It's poster number 3 and 4. Thank you for your attention. I think we have a little bit of time for questions. You did an awesome job, yes.